have been looking forward to coming to this conference. So, the work I'm going to present is not published yet, but hopefully it will be very soon. And it's a collaboration with Michael Proeti and Ole Christian Greiner Tollersrud. We are doing this project. For me, this collaboration has been really inspiring. And I was contacted 2-3 years ago to test the idea of Miguel and Ole. So, yeah, Washington is quite far from where we normally live. So Ole Christian lives in Tromsø and Miguel in Freiburg, and quite exactly between us, I live in Oslo. My background is in DNA repair and RNA editing, and that was also the reason why I was contacted. We have already heard about ADA1 and ADA2, and that there are major differences between these enzymes, except for one point that has not been mentioned yet. I think it's the pH optimum. Whereas ADA1 have a pH optimum of 7 to 8, ADA2 allows a lower pH of 5 to 6. We have touched on the extracellular localization, on the Km value, the high Km for ADA2 versus the low for ADA1, and also the adenosine concentration in blood, which in most ADA2 patients, I think, is normal. So that made us ask the question whether this generally believe that ADA2 is an extracellular protein. Maybe it's not the only role of ADA2. So, and also other hints gave us some ideas. For example, like as we have heard already, that interferon signaling is disturbed in ADA2, and that is often seen in interferonopathies where the nucleic acid sensing is defect. Moreover, if you go to the tree structure of ADA2 and compare it with ADA1, in ADA1 the active site is very narrow and small. This is the blue mesh, which is perfect for free adenosine. Whereas in ADA2, it's larger, like the yellow mesh, which may suggest that there is place for larger molecules, like DNA. Also, if we compare the amino acid sequence, so ADA1 and ADA2, ADA2 has an additional domain, which we often is referred to as the putative receptor binding domain, or PRB domain. This, if you model or test this, it may fit a DNA molecule. So, and finally, as has been already mentioned here, and also as we have found, that is ADA2 is localized in the lysosomes. And we know that the main function of lysosomes is to degrade macromolecules, like proteins, fatty acids, fat, yeah, but also RNA and DNA. And okay. So we wanted to test if ADA2 has affinity for DNA. We have already been demonstrated by Saviola that he also found this. So the test we do to check for DNA binding is to incubate a labeled DNA, radioactivity labeled DNA, and the protein, like the orange dots here, in a test tube for a while, and then run gels. And the free DNA will in these gels migrate along in the gel, and we'll see it in the bottom. And if we have complexes between DNA and protein, they will be on the top of the gel. So we did this, we purified human recombinant ADA2 from after expression in human cells, and tested binding. The first lane is only the DNA. It migrates in the front of the gel, and when we add ADA2, we get this smear. For me, this indicates that there are weak interaction between the DNA and the ADA2. The DNA here is single-stranded DNA, a short piece of DNA of 20 nucleotides. Then I tested the double-stranded DNA, and here the smear was gone. We tested different other substrates, like hairpin substrate. Here the DNA is double-stranded and have a small hairpin. Then we did see this weak shift on the top here. We tested another substrate where we removed the double strand, and we have only 2 single-stranded ends and then a double-stranded hairpin. And suddenly we got these strong and specific shifts. If the single-stranded ends were truncated, only having 7 nucleotides, the shift was gone again, indicating that ADA2 needs some size of the DNA molecule to bind. And also, when the single-stranded arms are longer and the double-stranded region is longer, the shift is really, really strong. So in our hands, the ADA2 proteins has highest DNA binding affinity when DNA binding affinity when the DNA has both single and double-stranded regions. And also, we tested the pH dependence of this binding. The first assay was done at pH 5.5, and we compared with pH 7.5, and also at pH 7.5, we still have some binding, although a little bit weaker, meaning that DNA in this circulation also can bind DNA. We tested some of the patient mutants, and of those we tested, the G358R protein did not bind DNA. This mutation is in the active site of ADA2. Then we was thinking, we have a protein that binds DNA. Could it also be that it deaminates DNA? So to test this, we set up an activity assay where we, again, uses a radioactively labeled DNA, incubate together with the ADA2 at pH 5.5. Which, if ADA2 is active, will make inosine in the DNA. An enzyme called endonuclease 5 is specific for inosines in DNA. It can bind, and what endo5 does, it cleaves the DNA downstream or on the 3' end of inosine, which means that we get a shorter product like here. And if we separate this on a denaturating gel, the full-length DNA will stay on the top, whereas the shorter products will migrate quicker in the gel. So in these assays, we are looking for bands on the bottom of the gel. So we incubated then DNA, a single-stranded DNA, and ADA2. So from left, only the substrate is on the top. only endonuclease 5 is also on the top because there are no inosines there, and the 5 does not do anything. And also only ADA2 is on the top because, yeah, it does not cleave. But if we have both ADA2 and endo5, we have this shorter product in the gel, which depends on enzyme concentration. And also, if we incubate over time, we get more product over time. And finally, if we test the pH, it's also dependent on pH and has an pH optimum at 5.5, whereas at 7 or 7.5, like it's in the blood, the deaminase activity is probably very low. We included ADA1 in these experiments, and ADA1 was not active on DNA. The sequence of the DNA I've used here has many adenosines, and we were interested to test which of the adenosines are cleaved or not cleaved, are deaminated by ADA2. We get only one band, so we thought that it's probably only one position. So to test this, we generated different oligonucleotides having adenosines in different positions. So this is the standard assay. Then we mutated adenosine number 6, and we still get the same band. However, if we mutate the first adenosine, A1, the band is gone, meaning that ADA2 prefers the first adenosine. Also, when we run them on a sequencing gel, we got this short product in the front of the gel, corresponding to 2 nucleotides, like the endo5 cleavage product. Yeah, and ADA2 is active both on single-strand and double-stranded DNA. This was the endo5 cleavage. And also in these experiments, we included the endogenous protein from pig brain. And also that has the same cleavage band as the recombinant human endo5. And also, single-stranded DNA is more often more efficient substrate than double-stranded, and also we find deamination at nicks and gaps in the DNA. We tested the mutants, and as for the DNA binding, the 358R did not deaminate DNA. I forgot to mention these bands, which we see in the pig brain ADA2, is probably because of co-purifying DNases in this preparation, according to the earlier presentation we had here. And then, okay, the way we detect the inosines here is the enzyme endonuclease 5, and we wanted also to test by a different method that it's indeed is inosine in the DNA. And to do that, we spotted the reaction, ADA2 DNA reaction on the membranes, and probed with an anti-inosine antibody. And those with red here has a 5' adenosine, which we have a signal here, here, and here. But we can see it for this substrate number 3, there's a really strong signal, and this has also a 3' adenosine in the other end, and it appears that ADA2 can deaminate adenosines in both ends. A third method to detect the inosine is mass spectrometry, and the same situation is seen here. The substrate with 5' adenosines give a signal, and the one having inosines in both ends give even a higher signal. So we have a protein that binds DNA, and it deaminates adenosines at either end. But the non-function of ADA2 is then the free adenosine. So to test the preference of DNA versus free adenosine, we did a standard assay with DNA, but then we added free adenosine altogether, and then the enzyme can choose. And if the enzyme chooses free adenosine, then the activity on DNA is expected to be low. But what we see here is that here is the standard reaction, where we have about 45% cleavage without adenosine. And indeed, we need to add 100,000-fold excess of free adenosine to see inhibition of the ADA2 activity on DNA. Which I think clearly shows that DNA is preferred above free adenosine for this enzyme. I will mention that free adenosine is what is used to diagnose DADA2 today. So this may possibly suggest that there are possibilities to improve. So I have told you that the ADA2 bind DNA, especially when it has both single-stranded and double-stranded regions. It could be that this is a mechanism to keep the DNA in the lysosomes. As was suggested before, it's in a way to compete with TLR9, it could be. Or it could also be to shield it or protect it from degradation by other enzymes in the lysosomes. Also, we have found that that ADA2 is deaminating DNA. But we yet do not know whether these two activities are concerted or they happen separately. And how these are regulated, we still do not know. And I want to remember in DNA, inosine is read like guanosine by cellular proteins. So this will make the DNA more G-rich, which could play a role, which I will show later. And also, I think this is the first example of a enzyme editing, doing A to I editing on DNA. And it could possibly be a tool for gene editing in the future. So then what is the biological function of these two activities? And with that, I say thank you for your attention.